We had the pleasure of presenting the Activ8 study, which was a randomized trial in first-line recurrent metastatic squamous cell carcinoma of the head and neck. It was a study that involved the use of a standard regimen in those patients called the extreme regimen, platinum, 5-FU, and cetuximab, and the experimental arm added a toll-like-receptor 8 agonist called motolimod. Toll-like-receptor 8 agonist has been in development for several years now, and what the early studies demonstrated was clear activation of the immune system, especially the innate immune system, natural killer cells, and myeloid-derived dendritic cells, which then allows activation and engagement of CD8 positive cytotoxic T cells. In a prior study, combined with cetuximab, the investigators were able to demonstrate safety and hints at early efficacy that combination may be quite effective. With all those preclinical and clinical data in mind, we set out to try and improve the standard treatment for patients with recurrent metastatic head and neck cancer by adding motolimod to the extreme regimen. We enrolled 195 patients over a span of about 2 years. The patient characteristics were typical of what we would expect in recurrent metastatic head and neck cancer, except for perhaps a slightly higher rate of HPV positivity, and that probably reflects the increased incidence of HPV positive in the United States. With that in mind, what we did not find was a difference between the two arms. Progression-free survival was the primary endpoint by central review, and there really was no difference between the control and experimental arm. The same held true if we looked at investigator review, no difference, and the same was true with overall survival. Having said that, what we did also see was that the agent was well tolerated with chemotherapy and with cetuximab, that we did see the typical toxicity associated with the drug of what we view as immune activation or toll-like-receptor 8 engagement. And in fact, the cytokine profile on both arms was reminiscent of prior studies and suggested that the toll-like-receptor 8 was being stimulated in the study. And so that began to get us to look at some of the other factors, especially with related to toxicity, that might be able to give us a signal. And what we found was that in patients who had an injection site reaction, so some real evidence that the toll-like-receptor 8 was being stimulated, well, those patients did benefit in the experimental arm, both with respect to progression-free survival and overall survival. So there is I think, very good clues that, in fact, we were hitting the target, that toll-like-receptor 8 can activate the immune system. Now we have to try and think about, well, who are the best patients? Who are the right patients to put on the subsequent studies? Because I think there is something there. So right now, the efforts are dedicated to trying to complete the data analysis. We're going to begin to look at other parameters of the immune system and immune activation and see if we can find a clearer signal with respect to patient selection. There is a study of the drug in combination with either a CTLA-4 or a PD-1 antibody, looking at perhaps compound immune investigation with combination therapy. That's what we call a window of opportunity study in patients with locally advanced disease, head and neck cancer, with biopsies prior to treatment and then on therapy, again, to try and get an understanding of what exactly is going on at the tumor microenvironment and hopefully lead us to the design of the next study with a, let's say, more appropriate patient population. The bottom line is the Activ8 study was a negative trial, but having said that, a lot of factors that lead us to believe that, in fact, this is a legitimate target and something to explore further.